Hello everyone, my name is Binay. Welcome to my channel LifeLure. So today we are going to learn about different variants of coronavirus which are related to COVID-19. So before going to detail of different types of variants, so we need to know the life cycle of coronavirus. So this is a coronavirus. So this is the membrane of coronavirus, and this is the RNA viral genome. This is the RNA viral genome, and these are the nucleocapsids, and this is the spike protein, which is very important for the binding of binding to the cells. So as I said in my last video, if you have not seen, please go to the link and check the video again. So two things which are very important for coronavirus are RNA viral genome and the spike protein. So suppose this is a cell of our lungs, and this is the SCE2 receptor. The spike protein of coronavirus binds to S2 receptor, and then the furin enzyme of the body of our body cleaves the spike protein, and then second comes to the TMP-RSS protease. It also cleaves the spike protein of coronavirus. Then the spike protein fuses with the S2 receptor, and then after that it gets internalized into the cell. So after internalization, the envelope opens up, and the viral genome, which is RNA in this case, comes out, and it makes different proteins which are related to the coronavirus, like spike protein, or maybe some other proteins like polymerase and some polyproteins. It makes proteins. Simultaneously, it also replicates its RNA using RNA dependent RNA polymerase. So after the replication, these RNA also make several capsid proteins, also envelope proteins, also spike proteins, and all together they make suppose this is the spike protein, nucleocapsid, and the envelope. And these proteins along with the RNA, they combine together, make more RNA viruses, and then they come out of the cell and they infect more cells. So this is the whole life cycle of a coronavirus, and in this life cycle there are chances that there will be some mutant viruses. So where are the chances? So first we'll know the chances, why the chances are there for the mutation. So because this is a RNA, which is the genetic material of the virus, so we'll make two considerations, DNA and RNA. So this is RNA and this is DNA. DNA is present in our body, but RNA is in coronavirus. So this RNA is a single stranded RNA, but DNA, what we have is double stranded DNA. So in this case, in coronavirus case, RNA makes new RNA through RNA dependent RNA polymerase. But in our case, this is the DNA, and the DNA replication is done by DNA polymerase. Here is the problem. So this RNA dependent RNA polymerase is very primitive one in the evolutionary process, but the DNA polymerase is very advanced. So this DNA polymerase has exonuclease activity. That means if there is a mistake in replication, if there is a mistake, suppose in this case, suppose this is A, T, G, C, suppose like this, and it replicates to A, and one mistake, suppose this is in place of T, there will be a C, G, C. So what DNA polymerase does, it goes back, cuts this nucleotide and then replace it with the T. That doesn't happen with RNA dependent RNA polymerase. So if there is a mistake, which happens in case of RNA dependent RNA polymerase, the mistake happens once in 10,000 nucleotide, 1 in 10,000 nucleotide, which is not replaceable. Not like DNA polymerase, it has no exonuclease activity. So if there is a mistake, the mistake stays. So suppose one example. So we will be talking about 501 mutation. So suppose 501 is a asparagine. What happens here? Asparagine H, G, A, and C, which makes, and this copies G, A, C, and after that the protein translation, which I told about here, protein translation. So here protein translation, after the translation there will be asparagine in this. Suppose this is the whole protein, here the asparagine will be there. Suppose in this process, RNA polymerase replication process, there will be a mistake. In place of G, it makes a U. There will be no change. The UAC will continue. So after this, the mRNA, this mRNA has UAC in place of GAC, and the next, when the virus infects another cell, the UAC will continue, and the UAC will always make, in place of asparagine, it will make tyrosine. So just remember that while we are writing, suppose this is 501 amino acid number, this is the amino acid number, and initially it was N. N is coded for asparagine, and it changes to tyrosine, which is Y. This is the single amino acid code. So this N to Y is important for the coronavirus. So now we'll learn about different variants of coronavirus. So first variant we saw was the UK variant, because it was seen first time in UK in 2020 February. So we called it as UK variant, and the scientific name, which is the nomenclature, different nomenclature was there. So that was pangolin nomenclature. So according to the pangolin nomenclature, it was B117. So after that, there were so many variants which arose in different countries. So people call them according to the country name. But because the many countries objected to this type of nomenclature, so WHO had a plan to replace this nomenclature, this country name, with alpha, beta, gamma, delta, like that. So the first one, which was found in UK, they called it alpha variant. So these are the mutations which are present in alpha variant. So 69 and 70 amino acid deletion. Suppose here is the 69 number amino acid and 70 amino acid which are present on S1 domain. Here 69 and 70, and these two amino acid deletion increase their infection rate. And another one which I already talked is N501 mutation. So this is the mutation which has strongest affinity with S2 receptor. Another mutation which is P681 mutation. So this is 681 is present on the furin cleavage site. Because of this mutation, furin, which is present on cell membrane, cleaves the spike protein more efficiently. So that helps the coronavirus enter efficiently into the cell. So because of these mutations, there will be increased in infection, which will increase in transmission. So because of these mutations, there are around more 80% more infection rate in these variants. And the second variant was the beta variant, which was first found in South Africa in May 2020, and it had major three mutations, which was first one 417K, 417N, 484K, and the same mutation which was already in alpha variant. So now what these two mutations do. 417N, which help in immune escape. Suppose there was a monoclonal antibody which different companies make to inhibit the binding of spike protein to the S2 receptor. So this escapes monoclonal antibodies 417N, K417N, and E484K. This also helps in escape the immune system, because till now whatever we have seen, the most antibodies which are produced in our body in response to the infection of coronavirus, they are made against this domain, receptor binding domain, and the mutation and the 484 present on this domain. That is why this domain mutation helps in escape the immunity of her. Suppose one person is already infected with coronavirus, and he or she, the person has antibody against this domain. Next time the virus comes which has this mutation, the antibody which was already produced will not recognize the coronavirus, and the coronavirus easily will escape the immune system and it will infect our body. Because of this mutation, this will show more transmission. So around this transmission rate was increased by 50% and reduction in immunogenicity, because the receptor binding domain, which is important for the binding and also immunogenicity, there was because of this mutation there. So this will help in escape the immunogenicity. And next variant is gamma variant, which was found in Brazil, and this type of variant was almost similar to the South African variant, which have similar mutations. This also had increased transmission and decreased immunogenicity. And the next one comes the Delta variant, which is now the most severe kind of variant. So now we will talk about the Delta variant, which was found in India first time. So it was found in October 2020. So initially the name was B16172. So then they named it as Delta variant, and this is the most unique type of variant, which has no relation with the previous variants. And the most important mutation of this variant is L452R. So we have seen this type of mutation happens in those people which already had infection and generated antibody or immunity against this virus. Suppose there's a person which was infected with the coronavirus previously, and the person has increased immunity. Suppose this is the antibody, and next time the coronavirus enters, this antibody will bind to the coronavirus, and this will not help the antibody will stop infecting the person. But because of this mutation in coronavirus, the binding of the antibody will not bind to coronavirus efficiently. So because of that, the coronavirus will infect the person easily. And the second mutation was the 4778 mutation, which is also helping. This is similar to 501, and this helps in binding to the H2 receptor. And another one is 681 mutation, which is present on furin cleavage site. So because of this mutation, there will be efficient furin cleavage, and it will fuse to the cell easily, and it enters the cell easily. So because of these mutations, this coronavirus variant has highest transmission efficiency, so which is around 200% more transmission rate than the previous variants. Now comes the Delta plus variant, which has all the mutations which were present in Delta, along with one more mutation, which is 417N, K417N. And this K417N mutation is present on the receptor binding domain of the coronavirus, and because of this mutation, it escapes monoclonal antibodies which are produced to block the infection. So it escapes class 1 and class 2 monoclonal antibodies. Simultaneously, this mutation, although this helps in escape monoclonal antibodies, there will be a little less transmission rate. In terms of escape to the immunity, this is more severe, but the transmission rate will be little less than the Delta variant. And probably that is the reason we do not see so many cases of Delta plus variants in our population as compared to the Delta variant. Delta plus variant may be the severe variant in terms of escaping the immune system. It has less transmission. So if we see in the third wave, my opinion is there will not be so many cases of Delta plus variant. If the third wave comes, there will be a new type of mutation in Delta variants, not the Delta plus variant which we see recently. So my opinion to stop these variants is we should make sure the sequencing of one cluster. If you see some clusters in our population, so first we need to make those sequencing and isolate those population from spreading more to different locations. So that will be the first prevention method. And my request is everyone should get vaccinated. Although the immunogenicity is reduced because of the Delta and Delta plus variants, still our body will respond to the coronavirus and it will give us protection. Thank you for watching my video. That is all about these variants, new variants. If you have any doubt, you can ask directly in my comment box. I'll answer those questions. If I find any more information about the new variant, I will make another video. To watch that video, please subscribe to my channel. And if you like this video, please like and share so that I can make more videos like this. Thank you. Bye bye.